Welcome to The Explainer. Today, we're diving straight into the wild world of bioinformatics to uncover exactly how scientists act as modern biological detectives. We're going to explore the science of phylogenetic tree analysis, which is basically this incredibly powerful tool used to reconstruct the shared evolutionary history of life on Earth.
Think about it. How exactly do scientists trace the origin of a deadly virus that's suddenly spreading globally? Or how do they figure out the common ancestor of vastly diverse species? Well, they don't just guess. To solve these biological cold cases, researchers use a highly specific map. Phylogenetic analysis is that ultimate map, allowing us to essentially rewind the evolutionary clock and trace the genetic fingerprints of life itself. Here is our roadmap for today's explainer. We'll be looking at Section 1, Mapping the Tree of Life, 2, Anatomy of a Phylogenetic Tree, 3, Steps to Build a Tree, 4, Computational detective work, 5, Real-world biological applications, and 6, The future of phylogenetics.
All right, let's jump right into Section 1, Mapping the Tree of Life. What exactly is a phylogenetic tree?
So what are we actually talking about here? Basically, a phylogenetic tree is a graphical representation of evolutionary history and the relationships among organisms, genes, or even proteins. It's built by analyzing molecular data, things like DNA, RNA, or amino acid sequences. Sometimes, if genetic material isn't available, like with ancient fossils, scientists will use morphological data instead. By comparing these sequences side by side, researchers can literally see how different species or genetic sequences have evolved from shared common ancestors.
Moving right along to Section 2, Anatomy of a Phylogenetic Tree, or Reading the Clues. Just like reading a physical map, you've got to understand the legend to know what you're looking at. A phylogenetic tree has a few distinct components. At the base, you have the root, which represents the most recent common ancestor of every single organism on that tree. As you move outward, you hit nodes. These are the branching points, kind of like forks in the road, that represent hypothetical common ancestors. The lines connecting everything are the branches, indicating the actual evolutionary lineage. In fact, branch length can even represent evolutionary time, or the sheer number of genetic mutations. And finally, you have the clade. A clade is a group of organisms that includes a specific common ancestor and absolutely all of its descendants. Now, these trees generally come in two different flavors. First, you've got your rooted tree. Because it has a defined common ancestor at its base, it gives us a chronological timeline. It shows a clear direction of evolution over time. The second type is an unrooted tree. This one completely lacks that ancestral origin point. It simply maps out genetic distance and how present-day organisms relate to one another, but we can't actually tell the direction of evolutionary time from it.
Okay, Section 3, Steps to Build a Tree. Let's look at the actual forensics procedure used here.
So, how do we actually build this thing? Well, it's a rigorous 6-step process. Step 1 is sequence collection, extracting DNA, RNA, or protein sequences from biological databases. Step 2 is sequence alignment, where scientists line these sequences up, almost like finding matching puzzle pieces, to see which regions are conserved and which vary. Step 3 is model selection. They have to choose a mathematical evolutionary model to describe how these sequences changed over time. Step 4 is tree construction, where the heavy computational lifting happens. Step 5 is tree evaluation, to statistically test if the tree is actually reliable. And Step 6, finally, is tree visualization, translating all that complex math into the branching diagrams we just talked about.
And just to be clear, scientists are absolutely not doing this with a pen and paper. They rely on a heavy-duty bioinformatics toolkit. We're talking software with names like Mega, which handles alignment and analysis. Others, like IQTree, PhyMel, and RaxML, step in for massive high-performance computing tasks and maximum likelihood construction. There are tools like Mr. Bayes that use advanced Bayesian probability models, and I found this fascinating. There's actually a software called Beast, which is designed specifically for molecular clock studies, helping determine exactly when in evolutionary time certain species diverged.
Which brings us to Section 4, Computational detective work. Let's talk algorithms and interrogation.
To find the correct evolutionary story, researchers have to pick their computational method very carefully. If a scientist needs sheer speed for a massive data set, they'll probably use neighbor joining, which is a lightning-fast distance space calculation. If they're looking at character-based data, maximum parsimony is great because it straightforwardly looks for the tree that requires the fewest total evolutionary changes. Then there's maximum likelihood, which brings in complex probability models to find a highly accurate tree.
And finally, Bayesian inference utilizes intense statistical probability models to provide high statistical reliability for the relationships. All right, so once we have our tree constructed, we have to ask the big question. How confident are we in this result? Enter bootstrap analysis. This is basically the ultimate confidence score for our evolutionary detective work. It runs the data through thousands of simulations to measure the reliability of every single branch on the tree, and it scores them on a scale from 0 to 100%. To put that into perspective, a score above 70% is generally considered good support. But if a scientist sees a branch with a bootstrap value greater than 90%, that is huge. That indicates very strong statistical support. It means we can be highly confident that this specific evolutionary relationship, that exact fork in the road, actually happened in nature. Let's bring this into the real world with Section 5, Real-World Biological Applications, or Case Closures. The impact of this science is quite literally everywhere, and it is a total game changer. In medicine, it's saving lives. We use it to track disease outbreaks and monitor how viruses are evolving in real time, which is absolutely crucial for assisting in vaccine and drug development. In biology, it helps us trace species evolution, classify microorganisms, and identify the common ancestors of all living things. And even in agriculture, tracing these genetic relationships allows scientists to drastically improve crop breeding and livestock genetics. But hey, let's be real for a second. No scientific method is completely flawless, right? Phylogenetic analysis definitely has its limitations. First off, the results depend entirely on the quality of your starting sequences. Bad data means a bad tree. Building these trees is also incredibly computationally intensive when you get into large data sets. Plus, phenomena like bacterial horizontal gene transfer, where bacteria basically swap DNA sideways with their neighbors instead of inheriting it vertically, can seriously complicate our ability to draw a clean, straightforward evolutionary tree. Finally, let's look ahead to Section 6, the future of phylogenetics. What is next for this field? This field is accelerating at a breakneck pace. Thanks to next-generation sequencing, or NGS, we're not just looking at single genes anymore. We're mapping entire genomes. Throw in cloud computing and high-performance computing, and our capabilities are exploding. But the part I find absolutely mind-blowing, artificial intelligence. AI-assisted phylogenetic inference is setting the stage for real-time global outbreak monitoring. Just imagine an AI predicting a pathogen's next evolutionary jump. All of this is slowly integrating with massive biological data sets to pave the way for true, personalized evolutionary medicine. So as we wrap up this explainer, it is super clear that phylogenetic tree analysis is undeniably one of the most powerful bioinformatic tools we have to look deeply into the shared history of life on Earth. But I want to leave you with this provocative thought. As AI maps our evolutionary past in real time, what will it predict about our future? Thanks so much for exploring this with us today, and keep questioning the science that shapes our world.